Singgalan ko Wilson. Calcium pectate. Calcium pectate CP. CPMLCP. Middle lamella. The Golgi cisternae are concentrically arranged near the nucleus with a distinct convex seesaw the forming phase and concave transfer maturing phase. forming phase and maturing phase. Cs, weeks. Distinct convex Cs are forming phases or convex, okay, and concave trans or maturing phase. Interconnected. They are entirely different. Forming phase. The Golgi apparatus principally performs the function. Okay, organelles are entirely different. But independent, the Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular target or intercellular or secreted outside the cell. Intracellular target delivered either to the intracellular target or secreted outside the cell. Delivered to the intracellular target or secreted outside the cell. Materials to be packaged in the form of vesicles or from the ER fused with the cis phase. Packaged in the form of vesicles from the ER fused with the cis phase and the Golgi apparatus and move towards the maturing phase. This is why Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasm are modified in the cisternae of Golgi apparatus before they are released from its trans phase. This is the important site of formation of glycoprotein and glycolipid. Pack material. Cs. Pack material. Golgi apparatus, packed material, cis and trans, cisternae. Intracellular, cellular, target deliver, deliver to intracellular target, pack material, cis and trans phase, vesicles from a modified and system delivered to intracellular target, secreted outside and secreted outside important site by fraction of protein and glycolipids. Okay. This is what is to be packed in the form of vesicles. Lysosome, suicidal bag, autolysis. Lysosome, suicidal bag, autolysis. Self-digestion. Okay. Suicidal bag, autolysis, self-digestion. Excuse me. There are membrane. These are membrane-bound vesicular structure. Okay. Lysosome के बारे में जो कुछ है बताएं. Lysosome. Plus bag, lysosome or suicidal bag, autolysis suicidal bag. Okay, lysosome, autolysis suicidal bag, different enzymes, DIFFER into different enzymes, ZYMS enzymes are present. Different enzymes are present. Hydrolytic enzymes. Hydrolytic enzymes. They're membrane bound organelles. These are membrane bound vesicular structure. These are membrane bound vesicular structure formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all type of hydrolytic enzymes, hydrolases, lipases, proteases, carbohydrates, optimally active at low pH, acidic pH. Yeah. Low. These enzymes are capable of digesting carbohydrate, protein, lipids, and nucleic acid. pH. Okay. These enzymes are capable of digesting carbohydrate, proteins, and nucleic acid. The isolated lysosomal vesicles have been found to be very rich in almost all type of hydrolase, lipases, protease, active at the acidic pH. The enzymes are capable of digesting carbohydrate, protein, lipid, and nucleic acid. Okay. Liquid pencil. Dry erase. ATM is equals to 1.01. Astaghfirullah. 1.01 10 to the power 5 Pascal. 5 Pascal. 10 to the power 5 Pascal. Yes to liquid condensation. Okay. Evaporation or vaporization. Solidification. Liquid fusion. Fusion, okay. Get a solid. This is gas to solid. Deposition. Sublimation. Solid to gas sublimation. Fusion. Solid to liquid fusion. Solid to liquid fusion. Okay. Liquid to solid. Solidification. Liquid to solid. Solidification. Liquid to gas. Liquid to gas. Sublimation. Liquid to gas. Sublimation. Gas sublimation. Gas to solid deposition. Okay. वो श्री ने ही कर दिया था हाइट वेट membrane-bound vesicular structure formed by the process of packaging in the membrane-bound vesicular structure formed by the process of packaging in Golgi apparatus. The isolated lysosome vesicles have been found to be very rich in almost all type of hydrolytic enzymes, hydrolases, lipases, proteases, carbohydrate, optimally active at the acidic pH. The enzymes are capable of digesting carbohydrate, protein, lipids, and nucleic acid. Even nucleic acid, okay? Carbohydrate, protein, lipids, and nucleic acid. Vacuoles. The vacuole is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory product and other materials not useful for the cell. The vacuole is bound by a single membrane called a tonoplast. Excuse me. In plant, single membrane called tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transfer of number of ions and other materials against concentration gradient into the vacuole. Against vacuole. Hence, the concentration is significantly higher in the vacuole than in the cytoplasm. Okay. In amoeba, the contractile vacuole is important for excretion in many cells, as in proteus. Food vacuole are formed by engulfing the food particles. Particles, food vacuole. Excretion को लगी amoeba में contractile vacuole. Contractile vacuole. Gas vacuole are found in blue, green, and purple synthetic bacteria. Those are gas vacuole, contractile vacuole, and food vacuole. Mitochondria, single mitochondrion, unless specifically stained, are not easily visible under the microscope. Physiological activity of the cell. Mitochondria Mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cell. Mitochondria per cell is variable depending on the physiological activity of the cell. In terms of shape and size also, considerable degree of variability is observed. Typically, it is sausage-shaped or cylindrical, having a diameter 0.2 to 1.0 micrometer, average 0.5 micrometer, and length 1.0 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure. With the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments. Each mitochondrion is a double membrane bound structure. Each mitochondrion is a double membrane bound. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane, okay, dividing its lumen distinctly into two aqueous compartments, that is outer compartment and inner compartment. The inner compartment is called the matrix. Inner compartment is matrix. The outer membrane forms the continuous limiting boundary of the organelles. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane. Outer membrane bio and the inner membrane bio. Okay. Astaghfirullah. Dividing the lumen distinctly into two aqueous compartments, that is the outer compartment and the inner compartment. And the outer compartment and the inner compartment. The inner compartment is called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The outer membrane forms the continuous limiting boundary of the organelles. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infoldings called the crista. The inner membrane forms a number of infoldings called the crista or cristae towards the matrix. Matrix, okay? Aqueous compartment, the outer compartment and the inner compartment. The inner compartment is called matrix. The outer membrane forms the continuous limiting boundary. The outer membrane forms the continuous limiting boundary of the organelle. The outer membrane forms the continuous limiting boundary of the organelles. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infolding called the crista or cristae or crista singular of the matrix. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Their own specific enzymes associated with the mitochondrial function. The mitochondria are the site of aerobic respiration. Mitochondria are the site of aerobic respiration with air. Okay, they produce cellular energy in the form of ATP. Hence, they are called powerhouse of the cellular energy in the form of ATP. They are called powerhouse of the cell. The matrix also possesses single circular DNA molecule, a few RNA molecule, and ribosome 70S, and the components required for synthesis of protein 70S ribosome in matrix of mitochondria. 70S RNA, few RNA, single circular DNA molecule, ribosome 70S. The mitochondria divide by fission. For the synthesis of proteins, the mitochondria divides by fission. Plastids are found in all plant cells, that is thylakoid, whole is granum. Plastids are found in all plants and in euglenoids, okay? Protista. Plastids are found in Papa ko bolu ka. Plastids are found in all plants and plastids are found in all plant cells and in euglenoids. These are easily absorbed under the microscope and as they are large. Plastids are found in all plant cells and in euglenoids. These are easily absorbed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plant. Based on the type of pigments, plastid can be classified into chloroplast, chromoplast, and leucoplast. Plastids are found in all plant cells and in euglenoids. These are easily absorbed under the microscope as they are large, and these are easily absorbed under the microscope as they are large. They bear some specific pigment, thus imparting specific color to the plant. Based on the type of pigment, plastid can be classified into chloroplast, chromoplast, and leucoplast. Chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis. The chloroplast contain chloroplast contains a chlorophyll and carotenoid pigment, which are responsible for trapping light and essential energy, light trapping light energy essential for photosynthesis. Chloroplast contain chlorophyll and carotenoid pigment, which are responsible for trapping light energy essential for photosynthesis. In chromoplast, fat soluble carotenoid pigments like carotene and xanthophyll and others are present. This gives the part of the plant a yellow, orange, or red color. Chromoplast. Chromoplast, fat soluble carotenoid pigment like carotene and xanthophyll and others are present. This gives the part of the plant a yellow, orange, or red color. The leucoplast are the colorless plastid. Leucoplast are colorless. The chloroplast contain chlorophyll and carotenoid pigment, which are responsible for chloroplast. Okay, chloroplast. Chromoplast means chloroplast contain chlorophyll and carotenoid pigment, which are responsible for trapping light energy essential for photosynthesis. That is chlorophyll. Chlorophyll. The chloroplast. The chloroplast contain chlorophyll and carotenoid pigment, which are responsible for trapping light energy. The chloroplast contain chlorophyll and carotenoid pigment, which are responsible for trapping light energy essential for photosynthesis. In chromoplast, fat soluble carotenoid pigments like carotene, xanthophyll, and others are present. This give the part of a plant a yellow, orange, or a red color. Carotenoid and xanthophyll, and other pigment. Carotenoid, okay. Chloroplast, chromoplast, fat soluble. Fat soluble carotenoid pigment. Fat soluble carotenoid pigments like carotene, xanthophyll, and others are present. This gives the part of the plant. The leucoplasts are colorless plaster, spheroid shape. Chloroplast, chromoplast, and leucoplast. Colorless plastid of varied shape and sizes which store nutrients. A myeloplast stores carbohydrate. Aleuroplast store protein and alluro and allylo stores oils, right? and fats, whereas aleuroprotein, okay. Amyloplast stores carbohydrate, starch, example potato. Aleuroplast stores oils and fats, whereas the aleuroplast stores protein. Aleuroplast stores protein. Myeloplast stores carbohydrate, starch, example potato. Aleuroplast stores oils and fats, whereas aleuroplast stores protein. Aleuroplast stores protein. Majority of the chloroplast of the green plants are found in mesophyll. Chloroplast of the green plants are found in the mesophyll cells of the leaves. Majority of the chloroplast of the green plants are found in the mesophyll cells of the leaf. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles, having variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number varies from 1 per cell of the Chlamydomonas और green algae Chlamydomonas protozoans में भी आता है to 20 to 40 per cell in mesophyll cells of leaf. Per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. Inner chloroplast membrane relatively less permeable. The space limited by the inner membrane of the chloroplast is called stroma. Okay, the space limited by the inner membrane of the chloroplast is called stroma. A number of organized flattened membranous sac called the membranous sac called the thylakoids are present in the stroma. Thylakoids are arranged in a stack like the pile of coins called grana, singular granum, or singular granum, or the intergranal thylakoid. Grana, granum, or intergranal thylakoid. Intergranal thylakoid. In addition, there are flat membranous tubules called the stroma lamella connecting the thylakoids of different grana. In addition, there are flat membranous tubes called the stroma lamella connecting the thylakoid of the different grana. The membrane of the thylakoid encloses a space called a lumen. The membrane of the thylakoid encloses a space called a lumen. The stroma of the chloroplast contain enzymes required for the synthesis of carbohydrate and proteins. It also contains small double-stranded circular DNA. Molecule and ribosomes. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoid. The ribosome of the chloroplast are smaller, 70s, than the cytoplasmic ribosomes, 80s. क्या हो गया है यहाँ पर? protein. One of the elaborate and process. Chloroplast contain enzymes required for synthesis of carbohydrate and protein. It also contains a small double-stranded circular DNA molecule and ribosomes. Small double-stranded circular DNA, double-stranded circular DNA, single circular DNA molecule, double-stranded, okay, double-stranded small circular DNA molecule and ribosomes. Chlorophyll pigments are present in thylakoid. Chlorophyll pigment की मांसा thylakoid मांसा, okay. And ribosomes of the chloroplast are smaller than the cytoplasmic ribosome. ATS, okay. Ribosomes, यहाँ पर भी तो ribosomes पढ़ा था, prokaryotic ribosomes या फिर क्या पढ़ा था, ribosomes ही पढ़ा था, और देखो 15/20 nanometer. Plasma membrane associated with plasma membrane. Gas vacuole. Ribosomes are the granular structure first observed under electron microscope as a dense particle by George Palade, 1953. They are composed of ribonucleic acid, RNA, and protein, and not surrounded by any membrane. Ribosomes are the granular structure first observed under the electron microscope as dense particle by George Palade, 1953. They are composed of ribonucleic acid, RNA, and protein. RNA is composed of ribonucleic acid, RNA, and protein, and are not surrounded by any membrane. Protein and ribonucleic acid and protein. They are composed of ribonucleic acid, RNA, and protein. They are not surrounded by any membrane. The eukaryotic ribosomes are 80S, while the prokaryotic ribosomes are 70S. Here, S. Svedberg's unit stand for the sedimentation coefficient. It indirectly is a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. Okay, the eukaryotic ribosomes are 80S, while prokaryotic ribosomes 70S. It stands for sedimentation coefficient. It is indirectly a measure of density and size. The eukaryotic ribosomes are 80S, while prokaryotic ribosomes are 70S. Here S. Svedberg's unit stands for sedimentation coefficient. It indirectly is a measure of density and size. S. Svedberg's unit stands for sedimentation coefficient. Density and size. Sedimentation coefficient, it indirectly is a measure of density and size. Composed of two subunits. साइटोस्केलेटल एलिमेंट ओके, यहाँ सीखते हैं. Cytoskeletal elements present in eukaryotic cell, plants and animals, both absent in prokaryotic cells. Cytoskeletal element present in eukaryotic cell, plants and animals, both present in eukaryotic cell, plants and animals. Okay, both absent in prokaryotic cell. Present in eukaryotic cell, plants and animals both, absent in cytoskeletal element present in eukaryotic cell, plant and animals both, absent in prokaryotic cell. Example, bacteria. Types of cell cytoskeletal element 3. Cytoskeletal element are of 3 types: microtubules, intermediate filament, microfilament, actin filament. Okay, microtubules, diameter 25 nanometer. 2 types of protein: alpha tubulin and beta tubulin. Present alpha tubulin and beta tubulin. 13 beta 13 alpha 13. One microtubule is equals to 13 protofilament. Microtubules formed by alpha and beta tubulin. Cytoskeletal elements are present in both plant and animal cell, but they have to be eukaryotes. They are absent in prokaryotic cell. Example, bacteria. Types of cytoskeletal elements: 3 types of cytoskeletal element. One is microtubules, other one is intermediate filament, microfilament, and actin filament. Microfilament or actin filament. Microtubule, intermediate filament, microfilament, actin filament. D is equals to 25 nanometer. Diameter is equals to diameter of microtubule is 25 nanometer. Intermediate filament diameter equals to 10 nanometer. Microfilament, actin filament, D is equals to 5 nanometer. Muscle contraction, amoeboid movement, amoeba and macrophage. Support nuclear membrane, intermediate filament, microtubules. 2 types of protein present: alpha protein and beta tubulin. Alpha tubulin and beta tubulin. Alpha tubulin, beta tubulin. Okay, one microtubule is equals to 13 protofilament. 13 protofilament. Microtubules formed by alpha and beta tubulin protein. Microtubules form structure. Flagella. Microtubules form structure. Flagella, cilia. Flagella and cilia. 9+2 arrangement of microtubules in exoneme of cilia and flagella. 9+2 arrangement of microtubules in exoneme of cilia and flagella. Basal body, centriole, basal body and centriole. 9+0 arrangement of microtubules, centrosome. Okay. Spindle fiber. Random horizon. Spindle fiber. No spatial arrangement. Spindle fiber. Random arrangement. 4 to 13 microtubules. 4 to 13 microtubules. 4 to 13 microtubules. Charge 2%. elaborate network of filamentous proteinaceous structure present. An elaborate network of filamentous proteinaceous structure present in the cytoplasm is elaborate network of filamentous proteinaceous structure, cytoskeleton. Cytoskeleton, proteinaceous filamentous structure present in the cytoplasm is collectively referred to as cytoskeleton. The cytoskeleton is a cell. In a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Maintenance of the shape of the cell, mechanical support, and motility. लाइट रिएक्शन ऑन थाइलाकोइड ऐसे नहीं याद होगा, मुझे ऐसे ही होगा, वैसे भी ये रिवीजन है। तो तुम्हें जैसे याद होगा वैसे तुम पढ़ो, मुझे जैसे याद होता है वैसे मैं पढ़ती हूँ। Present in eukaryotes, flagella. Present in both eukaryote and prokaryote, but their structure is different. Multicellular organism, unicellular organism. Cilia, cilium, and flagellum are hair-like outgrowth of the cell membrane. Hair-like outgrowth of cell membrane, okay? Cilia are small structure which works like oar, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for the cell movement. The prokaryotic bacteria also possesses flagella, but these are structurally different from that of eukaryotic flagella. In eukaryotic flagella, no hook, tubulin protein made up of tubulin protein. Okay, mitotic spindle composed of tubulin protein. Flagellin protein, okay. Tubulin protein. Eukaryotic flagella tubulin protein answer. Prokaryotic flagella, prokaryotic bacteria flagellin protein. Flagellin protein and tubulin protein. The electron microscopic study of a cilium or a flagellum so that they are covered with plasma membrane. Okay. The electron microscopic study of cilium or flagellum show that they are covered with plasma membrane. Their core, cilium or flagellum, okay? Their core, called the exome, possesses a number of microtubules running parallel to the long axis. The exome usually has 9 pair of doublet of radially arranged peripheral microtubules. 9 pair doublet radially arranged peripheral microtubules. The central tubules are connected and a pair of centrally located microtubules. 9 plus 2 array. The central tubules are connected each and is also enclosed by a central sheath which is connected to one of the tubules of each peripheral doublets by radial spoke. Thus, there are 9 radial spokes. Are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal body. Both the cilium and flagellum emerge from the centriole-like structure called the basal body. Centrosome and centrioles. Centrosomes is an organelle usually containing two cylindrical structure called centriole. They are surrounded by amorphous pericentriolar material. Both the centrioles in a centrosome lie perpendicular to each other in which each has an organization like cartwheel They are 9 plus O, made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the fibril is a triplet, and the adjacent triplet are also linked. The central part of the proximal region of the centriole is also proteinaceous and is called hub, which is connected with the tubules, which is connected with tubules of peripheral triplets by radial spoke made up of protein, which is connected with tubules of peripheral triplet by radial spoke made of protein. The centriole form the basal body of cilia or flagella and spindle fiber that gives rise to spindle apparatus during cell division in animal cell. Nucleus and microbodies. इतना करने के बाद I'll go and pray. Nucleus as cell organelle was first described by Robert Brown as early as 1831. Nucleus, Robert Brown. Okay? Later, the material of the nucleus stained by basic dye was given the name chromatin by Fleming. Nucleus was discovered by Robert Brown. Chromatin, given by Fleming. Chromatin by Fleming. Okay. The interphase nucleus of a cell when it is not dividing, has highly extended and elaborate nucleoprotein fiber called chromatin, nuclear matrix, and one of the more spherical bodies called nucleoli. Has highly extended and elaborate nucleoprotein fibers, okay. Has highly extended and elaborate nucleoprotein. The interphase nucleus has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli. Singular nucleolus. Nucleolus. Oh, same. 23, I guess. Same but different. What am I doing with my eyebrows? Extended and elaborate nucleoprotein fiber called chromatin. Has highly extended. Now I can't focus. I did that now. Has highly extended and elaborate nucleoprotein fiber called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli, singular nucleolus. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nanometer, called the perinuclear space, 10 to 50 nanometer, forms a barrier between the material present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bear ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of two membranes. These nuclear pores are the passages through which movement of RNA and protein molecule takes place in both the direction between nucleus and the cytoplasm. RNA and protein movement. Normally, there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. Example, teptum, coconut water, polar nuclei. Some mature cells even lack nucleus. Example, erythrocytes of many mammal and sieve tube cell of vascular plants. Some mature cells even lack nucleus. Example, erythrocytes of many mammal and sieve tube cells of vascular plant.
Would you consider the cells as living? Yes, I do. The nuclear matrix of the nucleoplasm contain nucleolus and chromatin. I have to do it. The nuclear matrix or the nucleoplasm contain nucleolus and chromatin. The nuclear matrix of the matrix or the nuclear matrix or the nucleoplasm contain nucleolus and chromatin. Nucleolus and chromatin. Nucleoplasm. Okay. The nucleoli are spherical structure present in the nucleoplasm. Nucleoli or nucleolus. Okay. They contain the content of nucleolus is continuous with the rest of the nucleoplasm, as it is not membrane bound structure. It is also the site for active ribosomal RNA synthesis. Nucleolus, nucleoli or nucleolus is site for active RNA synthesis. Large and more numerous nucleoli are present in cell actively carrying out protein synthesis. Chromatin genetic material. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fiber called chromatin. But during different stages of cell division, cell shows a structured chromosome in place of the nucleus. Chromatin contains DNA and some basic protein called histone protein and some non-histone protein. Study chapter, then revise them, make 30 minutes, so you will be strong. हाय सब लोगों को ज्ञान फेंकना है, मैं क्या करूं इतने सारे ज्ञान का. A single human cell has approximately 2-meter long thread of DNA distributed among the 4623 pair chromosome. 4623 pair chromosomes. You will study the detail of DNA packaging in the form of a chromosome in classroom. Every chromosome essentially has a primary constriction or the centromere. Primary constriction, centromere, on the side of which disc-shaped structure called kinetochore are present. Protein complex, okay, spindle fiber are attached kinetochore, centromere. Based on the position of the centromere, the chromosome can be classified into 4 types. The metacentric chromosome has middle centromere forming 2 equal arms of the chromosome. Metacentric, that is V. Metacentric, V-shaped, okay? Centromere. Submetacentric, L-shaped chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. Okay, this one is L. G. In case of acrocentric chromosome, the chromosome is situated close to its end, forming one extremely short and one very long arm. Acrocentric G. The telocentric chromosome has a terminal centromere I-shaped. Terminal I-G. A VL. This is I, this is J, VLJ. PQ, short arm P, Q long, okay. P and Q arm. Satellite, not present in all chromosome, role in nucleolus formation. Satellite DNA has role in nucleus formation, non-staining. Secondary constriction. Interesting one. Sometimes a few chromosomes have non-staining secondary constriction at a constant location. This gives the appearance of a small fragment called the satellite. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plants and animal cells. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. Okay? Microbodies. इसके एग्जाम में पेरोक्सीजोम्स, प्लांट सेल, एनिमल सेल. Peroxidase enzyme. Catalase and peroxidase. Let me clear enzyme. Microbodies. Microvilli, plasma membrane, centriole, peroxisomes, microbodies, lysosomes, ribosomes, mitochondria, rough endoplasmic reticulum, cytoplasm, nucleus, nucleolus, active RNA synthesis, nuclear envelope, smooth endoplasmic reticulum, Golgi apparatus, chloroplast, chromoplast, plastid, cytoplasm, peroxisomes, microbodies, microtubules, cytoskeletal element, plasmodesmata, smooth endoplasmic reticulum, middle lamella, calcium pectate, smooth endoplasmic reticulum, lysosomes, rough endoplasmic reticulum, nucleus, nucleolus, Golgi apparatus, nuclear envelope, plasma membrane, vacuole, middle lamella, calcium, magnesium pectate, cell wall, mitochondria, ribosomes. Who is this curry? Cell the unit of life, biomolecules, cell cycle and cell division. Okay, biomolecules पढ़ लेते हैं ना इससे पहले। फिर से आगे एक बात तो करूँगी कि biomolecules, this one is biomolecules, right? Classification and nomenclature. Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Classification and nomenclature of enzymes. Thousands of enzymes have been classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the type of reaction they catalyze. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on types of the reaction they catalyzes. Enzymes are divided into 6 classes, each with 4 to 13 subclasses. Enzymes are divided into 6 classes, each with 4 to 13 subclasses, and named according by a 4-digit number. ये 4-digit नंबर वाला समझ में नहीं आया, लेकिन क्या ही कर सकते हैं। ऑक्सीडो रिडक्टेज और डिहाइड्रोजनेज, ट्रांसफरेज, हाइड्रोलेज, लाइज एंड आइसोमरेज along with ligase. Dehydrogenase, oxidoreductase, dehydrogenase, oxidoreductase, dehydrogenase, oxidoreductase, dehydrogenase, reductases. We have dehydrogenase, oxidoreductase, dehydrogenase, ligase है ना, आता देखिए सर, transferase. It's okay, Ima. Transferase, hydrolase, HYDRO, hydrolases, and ligase, LYGASES, and isomerase. Wait a minute, isomerase. Just wait. What the heck is wrong? You bro. I don't know. Shut up. कुछ ना कुछ
physical chemistry. Classification of nomenclature and enzyme. Classification of nomenclature and nomenclature of enzyme.
Oxidoreductive. It catalyzes oxidation and reduction reaction. An example is oxidase and dehydrogenase. Second is the transferase category. It catalyzes the transfer of groups from one molecule to another molecule. An example is kinase and phosphorylase. Third is the hydrolase. Hydrolase catalyzes the addition of water. And examples are phosphatase and peptidase. उसके बाद lyase. It catalyzes the breaking of CC bond, carbon oxygen bond, and carbon nitrogen bond. And examples are synthase and aldolase. Fifth is the isomerase. It catalyzes the intermolecular rearrangement. And examples are mutase and epimerase. Last is ligase. It catalyzes the formation of carbon oxygen bond, carbon nitrogen bond, and carbon carbon bond. And example is carboxylase. Today I am going to discuss about the classification of enzymes. There are 6 classes of enzymes. First is the oxidoreductive. It catalyzes the oxidation and reduction reaction. An example is oxidase and dehydrogenase. Second is the transferase category. It catalyzes the transfer of groups from one molecule to another molecule. आंसर इज हेलिकेज। लेकिन अगर मैं वही बात करूं कि ट्रांसक्रिप्शन के दौरान अनबाइंडिंग ऑफ डीएनए हेलिक्स कौन से एंजाइम करता है, द आंसर विल बी आरएनए पॉलीमरेज। सच सच बताइएगा, आप में से कितने बच्चों को ये डिफरेंस पता था? ड्यूरिंग डीएनए two strands of DNA are unbinded by helicase enzyme, whereas during transcription the two strands of DNA are unbinded using RNA polymerase। इसी तरीके के कुछ रिवीजन वीडियोस के लिए स्टे ट्यून्ड टू
Nomenclature and nature of enzyme. Classification and nomenclature of enzyme. Mnemonics. Trick to remember enzyme classification. Okay.
Remember the main 6 classes of enzymes. Trick it is auth, lead. O for oxidoreductase, T for transperases, H for hydrolases, L for lyases, I for isomerases, L for ligases. First is
reductase, oxidoreductase or dehydrogenase. Dehydrogenase. Transferase, hydrolase, 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 ligase, isomerase, and lipase,
lipase, lysis and ligase, lysis, lysis, ligase.
To remember the main 6 classes of enzymes,
एक सवाल पूछूंगी और इस वीडियो के कमेंट सेक्शन में सच सच बताना आप में से कितने बच्चों को इसका आंसर सही पता था। ड्यूरिंग डीएनए रेप्लिकेशन, अनवाइंडिंग ऑफ डीएनए हेलिक्स इज डन बाय व्हिच एंजाइम? द आंसर इज हेलिकेज। लेकिन अगर मैं वही बात करूं कि ट्रांसक्रिप्शन के दौरान अनवाइंडिंग ऑफ डीएनए हेलिक्स कौन से एंजाइम करता है, द आंसर विल बी आरएनए पॉलीमरेज। सच सच बताइएगा आप में से कितने बच्चों को ये डिफरेंस पता था। During DNA replication, the 2 strands of DNA are unwinded by helicase enzyme, whereas during transcription, the 2 strands of DNA are unwinded using RNA polymerase. इसी तरीके के क्विक रिविजन वीडियोस के लिए stay tuned to Biology.
Concentration of substrate. Okay, concentration of substrate. With the increase in substrate concentration, the velocities are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzyme molecules to bind with the additional substrate molecule. Figure 9.7. Figure 9.7. Okay, of substrate enzyme activity, it rises, and after Vmax does not rise further activity. The activity of enzyme is also sensitive to presence of specific chemicals that binds to enzymes. When the binding of the chemicals shuts off enzyme activity, the process is called inhibition, and the chemical is called inhibitor and inhibition. Okay, when the inhibitor closely resembles the substrate in molecular structure and inhibits the activity of enzyme, it is known as competitive inhibitor. Also called substrate analog. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind, and as a result, the enzyme action declines. Substrate cannot bind. Okay. Example: inhibition of succinic dehydrogenase by malonate. Which closely resembles the substrate succinate in structure. Malonate, succinate. Okay. Such competitive inhibitors are often used in the control of bacterial pathogen. Of substrate. Graph. This is time. Okay, substrate concentration. V max. Okay. V by 2. V max upon 2, right? Km value. Michaelis constant. Km. Michaelis constant. Okay. Substrate concentration of substrate. As we increase substrate concentration of substrate, we increases. Okay. Competitive inhibition. Example of competitive inhibition. एग्जाम्पल ऑफ कॉम्पिटिटिव इनहिबिशन क्या होता है? ओके, इनहिबिशन ऑफ सक्सिनिक dehydrogenase, inhibition of dehydrogenase, MALAT, malate, resembles SUCCI, succinate, astaghfirullah, okay? Succinate, inhibition and inhibitors. Reasons. Oxidoreductase dehydrogenase transferase. Ligase, lipase ligase, lysis, is GESES. Isomerase L. Othlen Hydrolase. 6 classes, 4 to 13 or 4 to 16, 4 to 13 subclasses. 4 to 13 subclasses. 4 to 13 subclasses. Okay. Activation energy without enzyme. Potential energy progression of reaction. This is exothermic, endothermic, exothermic and endothermic. I'll go for class, okay? I'll come at 6:30 time. Bye. I'll go now. Shri, are you there? Shri, are you there? I don't think you are there. Shri Bhai Sahab, oh Shri Bhai Sahab, are you there? I think he left. Okay. Bye all. I have to go now.